Welcome. Today's second session of the Science Afternoon will be discussions of phenotyping new MPS II variants. This is a critical question as we move forward, hopefully soon, to getting newborn screening approved on the RUSP panel for MPS II. We have two talks today, one from Dr. Richard Steet at Greenwood Genetics Center and one from Dr. Patricia Dickson at Washington University in St. Louis. These are both based on National MPS Society funded research, and I know you'll be very excited to hear these presentations. Our first presentation from Dr. Richard Steet looks at zebrafish models and their ability to help us predict mutation and phenotype in both MPS I and MPS II. Thank you again and enjoy the presentation.
I want to thank the organizers of this conference for the opportunity to share with you our efforts over the past year developing an experimental platform that we can use to predict whether IDUA or IDS variants are going to be causative of MPS I and MPS II. Just to give you an outline of this talk, I'm going to start with an overview of newborn screening for MPS disorders and highlight how this newborn screening is leading to the increased identification of novel gene variants in different MPS genes. And then we'll talk about a problem faced by a lot of families in which the newborn screening and any of the follow-up testing is really inconclusive. And this leaves them in a situation where they have to wait and see whether or not an MPS, signs of an MPS disorder will develop. And I think we can all appreciate that's problematic in terms of thinking about when to initiate any sort of therapeutic intervention. I'll also talk about another problem that's arisen out of newborn screening, and that's an issue that we call pseudodeficiency. Then we'll move into our efforts to developing this platform and how this platform allows us to determine a value we call relative specific activity. And what we think the value of that relative specific activity is in terms of being able to stratify and understand whether these variants are going to be disease causing. Then I'll share with you the results on an initial set of IDUA variants and how we can use that information again to stratify these variants. And we'll talk about their predictive value and finish the talk with a somewhat rare situation where we have two variants that are present on one copy of the gene and how this same platform can be used to help us resolve that situation. Just to ensure that we're all on the same page in terms of nomenclature, I just want to review some terms that I'm going to use throughout the talk. I will use the term variant, and that simply refers to mutant or mutation within a gene, a change in the DNA, the sequence of DNA. Individuals with MPS I will inherit one variant from each parent. Situation with MPS II, because it's X-linked, is a little bit different. Cis variants are that situation where two variants are found on one copy of the gene. So we refer to those variants as being in cis. Pathogenic just refers to disease causing. Pseudodeficiency, which I'll review in more detail, is a situation where enzyme activity is read as low, but not low enough to be disease causing. And then a term that you'll hear again throughout the talk is this specific activity. Specific activity just refers to the activity of an enzyme relative to the abundance or the amount of enzyme that is there. So newborn screening for MPS disorders is expanding across the US. With its addition to the recommended uniform screening panel, MPS I is screened for in numerous states, and at least two other states have now added MPS II, Missouri and Illinois. We know that newborn screening is important because if we can resolve or identify patients that will develop MPS disorders and there are therapies that are available, we can start those therapies early. And the early use of therapies leads to better outcomes for patients. So the sooner we understand whether the condition is there, it gives the opportunity to initiate the treatments as early as possible. And this helps to improve the outcomes for the patients. Newborn screen is just measuring enzyme activity from a dried blood spot. The activity that one might get could be out of the normal range. This could indicate a positive MPS diagnosis, but typically we do other tests in order to confirm the screening result. These tests would include looking for elevated levels of sugar polymers called glycosaminoglycans or GAGs within urine or plasma. This can help be supportive and provide clues towards the diagnosis. And a lot of times an individual's DNA is sequenced after a positive newborn screen to look for variants in any of the MPS genes. So the more newborn screening that we do, the more we're finding new IDUA and IDS variants through this process. Many patients with a positive newborn screen, unfortunately, will also have unclear follow-up testing. In some cases, if the DNA sequencing identifies a variant that we've already run into, this can help confirm or exclude a diagnosis. But many times, the variant that is identified through the DNA sequencing is novel. That means it has not been seen or studied in the lab before. And we call these variants of uncertain significance, or VUS. So if you have a positive newborn screen, unclear secondary testing, and then the DNA sequencing identifies a VUS, this really leaves people with a stressful and concerning situation. They have to wait and see. The current information doesn't allow them to confirm a diagnosis. So a lot of times they're told, let's wait and see whether there are any clinical signs of an MPS disorder. But this waiting and seeing delays any treatments because we simply can't confirm the MPS diagnosis. So how about this problem of pseudodeficiency? Sometimes a newborn screening will give an enzyme result that is low. It's below the normal range. It may even be into the affected range. But we know after the DNA testing that then we may identify a variant that we know is not disease causing. So despite the reading being low, we have the experience to say that the identified variant won't be disease causing. And that's this scenario that's referred to as pseudodeficiency. Those variants are referred to as pseudodeficient variants. So like VUS, pseudodeficiency will create uncertainty until we can confirm that that low enzyme reading that was picked up during the newborn screen is not actually going to lead to an affected child or individual. So both of these things, the VUS and the pseudodeficiency variants, can be addressed if we can essentially test how each of those individual gene variants affects the function of the enzyme that is made by that gene. So we're essentially saying, let's make that form of the enzyme and then do some tests and ask what exact impact does it have on that enzyme? And I'll review the platform that we used initially. So the way we did this is we made a virus or viral particles that either carry the DNA sequence, the normal IDUA sequence, or the variant IDUA sequence. We take those virus and we use them to infect cells that lack any of their own IDUA. So the only IDUA that's being made is what we introduce by the virus. And then we take those cells and we do an enrichment step where we remove all the cells that aren't expressing any IDUA. And we end up with an enriched cell population that's either making the normal IDUA enzyme or the variant IDUA enzyme. And then we can go and do some experiments with these cells. And we can ask a couple different things. First, the one parameter is how much of the enzyme is being made and expressed. And we can do this using an antibody against that enzyme in a technique we call a Western blot. That will allow us to quantify how much total enzyme is there. And then we can do on the same cell population an activity assay And this will allow us to determine the activity of the enzyme expressed by IDUA in those cells. We do this using fluorescent substrates to that enzyme. Then we take these two pieces of information, the activity of the variant enzyme and the amount of the variant enzyme, and we calculate this ratio that we call the specific activity of the variant enzyme. Once we get this value, what can we do with it? Well, we can compare it to what we get with the normal or wild type version of the enzyme. And it's that value that we determine that is the relative specific activity. It is how much specific activity in the variant form of the enzyme is there relative to what you would see if you had a normal copy of that enzyme. So in order to test and optimize our platform, we chose an initial set of IDUA variants. This included several variants that were known to be pseudodeficient. These shown here, these four. And what you're looking at here is just the change, the amino acid within the protein sequence that's changed to another amino acid. And that's just a common nomenclature that we use in the field of genetics. We also included a known pathogenic allele that's associated with Hurler, this one here, and then two other pathogenic alleles or variants that are associated with a more attenuated form of MPS I called Shea. And then lastly, we included two variants of uncertain significance. These are variants where there's no experimental evidence to classify them as either pathogenic or pseudodeficient. And this would be a good test of whether the platform could help us resolve those two VUS. So this is what the results look like. Here's the enzyme activity results over here and the enzyme amount results over here. Shown here in the first lane is the wild type. We set this to 100, and then we get a relative amount of enzyme activity for all the variants that we tested. And I think you can appreciate right away how much lower these are compared to the wild type. But one question would be, and this is why both measurements are important. One question would be, are these low because you're not making any enzyme at all? Or are they low because the enzyme just isn't as active? And that's why we do this second test to look at amount or abundance. What you can appreciate is all of these do actually make the enzyme. They do make different amounts of it. But we found some other interesting features. For instance, here with the wild type, we're able to pick up these processed forms of the enzyme in wild type. And also with this Hurler variant, we see some processing. But many of these other ones, including the pseudodeficiency variants, don't seem to show us the processed form of the enzyme. We don't know what that means, but it might be an interesting clue. The other thing we do throughout this platform is we test two different cell clones that we generate from our different cell populations for each variant. So we can compare what the specific activity will look like in two different clones as we go through this process. So then we take that information and we generate the relative specific activity for each IDUA variant. So again, this is going to reflect the amount of enzyme activity over the abundance of the enzyme that is there. And this is the type of results we get. You get a number. So this one, for instance, this histidine at position 82 to a glutamine is going to be 26% of the wild type in terms of its specific activity. And you can see the values for these other ones. Clearly this one, which is the Hurler, is significantly lower, several orders of magnitude lower than any of these other ones. And then several of these, while low, still do retain a decent amount of specific activity within our experiments. So here's another way of looking and summarizing these results. This line right here, this is the specific activities plotted on a log scale. This line right here represents 1% of the wild type activity. So everything above this line will be greater than 1%. Everything below will be less than 1%. These do not represent negative values. They simply show that they're below that 1% threshold. And then we look at each different variant, right? We take the specific activity we got from the 2 clones and average those, and that's what's plotted here. So you can see, yes, all of these ones that we call pseudodeficiency variants are lower than wild type, but to variable levels. We have some that are only 10% have the specific activity that's 10% of wild type, whereas others are much higher. And then we can see here, here's our 2 Shea variants. Again, they're lower than any of the pseudodeficiencies, but they're not as low as this Hurler. And we even get more specific information. There's actually quite a distinction between these 2 Shea variants that I think is important to consider when you find these in combination with other variants. Now, how did we do in terms of the 2 VUSs? So these are shown right here, right? This one and this one. And these clearly fall in that pseudodeficiency range. So at least when we're looking at these on their own, I think we can have confidence that these are actually pseudodeficiency variants like these other known ones that are in that group. So this other scenario, and I think we feel like we can use this platform to address this scenario of what happens when you have IDUA variants in cysts. Now we know for MPS I, patients have 2 IDUA variants, one from mom, one from dad. Both of these really may require functional testing in order for us to have full confidence of a diagnosis. But occasionally we also run into a situation where DNA sequencing reveals 2 variants on the same copy of a gene. And that's what's shown graphically down here. So here's a situation where, in this case, the mother has 2 different pseudodeficiency variants on one copy of her IDUA gene, and dad has one copy of a pathogenic variant on one of his genes. So both of these would be considered carriers. However, if the child inherits this copy from mom and this copy from dad, they will have this resulting genotype where they have one copy of the gene with a pathogenic variant, the other copy of the gene with 2 pseudodeficiency variants. And we really don't know whether this child would be affected, mainly because we don't know what the effect of these 2 pseudodeficiency variants is in combination. And that's where we think our platform can be useful. So we can take that copy of IDUA, including the 2 pseudodeficiency variants, introduce it into our cells, and do the same types of experiments we did before. Ask how much enzyme is being made, what is the activity of that enzyme, and then determine a relative specific activity. And the real question is, to what extent is that combination going to be additive? In other words, maybe it reduces the activity of that version of the enzyme more than the single variants would, but not enough to push it into the pathogenic range. However, anytime you have 2 variants, they may have a synergistic effect or a combined effect that's worse than the combination of the 2 individual ones. And I think that being able to use this platform is important to resolve that question. So we are currently testing 2 combinations of known pseudodeficiency variants and to really resolve and answer this question of what happens when you have 2 pseudodeficiency variants. Do they combine to make a pathogenic copy of that gene? So I hope I've convinced you that we have established a useful new biochemical platform that we can use to determine a value called the relative specific activity of variant IDUA encoded enzymes. This value is helpful because it allows us to classify variants that are identified through DNA sequencing as either pathogenic or pseudodeficiency, and it allows us to stratify these and sort of line them up and determine which ones might have a more severe impact on the function of the enzyme. Importantly, we can use this to also determine the effects, again, a somewhat rare situation when 2 variants are found on the same copy of one gene inherited from either parent. We are currently optimizing our platform to make it faster and be able to answer this question more quickly. We're also expanding our work into looking at the IDS variants as well. But I think ultimately we would like to see a platform for every MPS disorder so that when newborn screening is done and secondary testing and DNA sequencing lead to inconclusive results, we have a way that we can apply to help address and confirm or to not confirm a diagnosis of MPS. So I want to close by acknowledging first Dr. Suk-Ho Yu, who is a staff scientist on our team, who's really been instrumental in all the work that I've shown you and is continuing to help develop new platforms. And also to acknowledge Drs. Heather Flanagan Steet, Laura Pollard, and Tim Wood, who have helped encourage the development of this platform and really seed some of the ideas and necessity for this platform. And have been wonderful colleagues, and we're anxious to continue our work with them. I also want to acknowledge the support of the National MPS Society that allowed us to develop the platforms for IDUA and IDS, as well as all the funders of our research on lysosomal storage disorders. And with that, I will stop and look forward to taking questions and to the discussion.
Thank you, Rich, for such an important topic that builds on so many of the issues and so much of the information that's been presented today and in the previous days of this conference. Indeed, being able to provide a better understanding of future phenotype based on results of newborn screening is such a critical issue that the National MPS Society was keen to be able to fund 2 projects that looked at basically the same problem from different angles. And one of those was the presentation that you will hear from Dr. Patty Dickson that really covers a similar aspect of MPS II disease, defining phenotypes, but using a very different approach. So without further ado, I will let the talk roll from Dr. Patty Dickson, and keep those questions coming because we'll have a live discussion afterwards. Thank you and enjoy.
Hello, I'm Patty Dickson, medical geneticist at Washington University in St. Louis. I'm going to share my slide. Okay, hopefully that's sharing the right slides for you. So I'm going to talk to you today about the classification of IDS gene variants using saturation genome editing. IDS is the gene for Hunter syndrome, iduronate 2-sulfatase. Hunter syndrome newborn screening is being conducted in some states, including Missouri, using dried blood spot assay for iduronate 2-sulfatase. And a low level of enzyme could mean MPS II, or it could mean something called a pseudodeficiency, which means that the enzyme is low, but there is no disease. In order to determine whether a newborn who tests positive with a low level of iduronate 2-sulfatase might have MPS II, there are 2 confirmatory studies that are generally performed. And this has changed a little bit, but it currently still these 2 may be performed. So one is an IDS gene testing where they sequence the IDS gene to look for whether it has any mutations. However, the problem is that there are many what's called variants of uncertain significance. You've probably already heard about this from Dr. Steet. And so we don't know how to interpret the genetic testing a lot of times. And then the other confirmatory test, which is probably the most reliable, we think, is glycosaminoglycans. And so you probably know about urinary glycosaminoglycans. There's also the ability now to test glycosaminoglycans in blood. There is the ability to test glycosaminoglycan levels in dried blood spots now, which is being done as now a 2nd-tier test to prevent babies from being called back if they test low for iduronate 2-sulfatase, but then have a normal glycosaminoglycans. We don't expect people who have normal levels of glycosaminoglycans to have MPS II, but we really don't know very much about the levels of glycosaminoglycans at birth, even today, in people who have MPS II. So an illustration of the problem with newborn screening and predicting these variants happened as my first case of positive newborn screen for Hunter syndrome relatively shortly after I moved here to Missouri. So we had a newborn screen that came back and was handled by the office. It was a low iduronate 2-sulfatase activity, and they test the other sulfatases because there's something called multiple sulfatase deficiency. So they have to make sure that all the other sulfatases look okay. So those were all fine. And then they called this baby back in for just a test and did the repeat assay, which was low again. And then they did urinary glycosaminoglycans. But these were normal, but they were in the upper limit of normal. They also did molecular testing for the iduronate 2-sulfatase gene, and it found a variant, a missense variant that had not been reported before. But the laboratory the commercial laboratory that was doing the testing called the variant likely pathogenic. And when I asked them about this later on, why they called it likely pathogenic, they said it was because of the biochemical results. So because the lab report classified the variant as likely pathogenic, the family was counseled as likely Hunter syndrome and scheduled to see me in clinic. So I saw this baby. I think he was about a month old at the time. And I looked over all of the results and was pretty surprised by the commercial laboratory calling the variant likely pathogenic based on a normal glycosaminoglycan result. I later learned that the American College of Medical Genetics their classification system doesn't really work very well for inborn errors in metabolism, where relatively damaging mutations that cause low levels of enzyme are actually pretty well tolerated. And so you can have a very low level of enzyme and a damaging mutation that through all of their prediction tools predicts that it's damaging to the protein, yet it doesn't actually cause MPS disease. So this baby had normal urinary glycosaminoglycans and had normal, what I sent a non-reducing end assay, which is an assay that's fairly specific for mutations that remove the enzyme that's needed to cleave a certain end off of the glycosaminoglycan. So if you're missing iduronidase, it'll detect a certain iduronic acid residue at the end of that glycosaminoglycan, and that's offered commercially at a laboratory, and that was normal. And we were able to close the case, and the baby does not affected with MPS 2. So to try to solve this problem, I partnered with the laboratory of Christina Gurnett here at Washington University. And she and her group had just recently developed a technology for creating systematic mutagenesis library. In other words, creating a large number of variants in a library, what we call a library in the laboratory, that could be done relatively quickly. And it's done with something called reversibly terminated inosine. So inosine is a base that partners with any of the other bases, A, G, T, C. And so they're able to generate, using this inosine, a rapid library of any different variant in the coding region of a particular gene. Then they amplify and can precisely generate these single mutations in this sequential fashion. And so they had demonstrated this in a beta-lactamase gene, testing 800 possible variants by using this method within a 200 base pair region of the active site of this gene. And then they found that they were able to classify the variants by abolishing, how they abolished ampicillin resistance to define the active site. So they were able to show that they could do this in an efficient way. And so the idea behind being able to do this for iduronate 2-sulfatase is that if we could create a library of all the variants in the IDS gene, have an assay that would identify the affected versus the unaffected cell population, and then on a way that was sortable by a fluorescent sorter. And then you could sort those cells into bins and classify them as by sequencing to determine what variant you found in each of the bins. And the Gurnett lab has since demonstrated that this could be done for a variety of other genes. For example, this is an assay that they were able to sort cells with SLC2A1 variants, showing that they had variants that could impact growth negatively versus not. And they had a GFP reporter line for SMAD3, and they were able to sort to determine the correct genotype off of their SMAD3 reporter line. So we generated a Tillerone experiment to see whether this might be feasible in MPS II. So for the Tillerone experiment, we took this hap1 cell line that the Gurnett lab liked to use. It's a haploid cell line, and we incubated it with a substance called Tillerone. The Tillerone has been around for a long time, and you add it to cells and it mimics MPS by binding to the glycosaminoglycans and keeping them retained in the lysosome so that they accumulate to the extent of MPS. And they actually accumulate glycosaminoglycans that are about 3 or 4 times the wild type level, which is pretty comparable to what you see in a liver cell, for example, of mice with MPS I. So these were then stained for lysosomal associated membrane protein, LAMP1, with a fluorescent antibody in order to determine, to generate the fluorescent signal, and so that we could see if this could be something we might be able to sort on. And we expected to see a larger fluorescent signal in cells that were incubated with the Tillerone, because we generally see more LAMP signal in tissues of MPS affected mice, for example, than in wild type. So this is the initial result that we generated. So the Tillerone was on the right and no Tillerone on the left. And the staining in red is for LAMP. The staining in blue is just a nuclear stain. So here you can see the little red areas on the cells that were stained, that were incubated, with Tillerone. And then on the left, you see the wild type cells that don't really have much red signal. So we proceeded with support from the MPS Society to an IDS knockout experiment. And the Gurnett laboratory generated a knockout line for idurate for IDS. It was an exon 9 deletion in the hap1 cell line, and we've subsequently determined that it has no idurate 2 sulfatase activity. And these were cultured and sorted by flow cytometry. However, as we worked on this line with the Gurnett laboratory, we were not able to sort based on the LAMP1 signal alone. It was just not reliable in distinguishing the MPS II line from the control cells. And so in trying to determine what to do next, we turned to a colleague here named William Buchser, whose laboratory works with something called the cell raft system. And this is a small grid where you can culture single cells actually in each of these wells. You seed them, you can image them, and then if you want to release them for isolation and analysis, the raft will allow you to lift the cells, it can be collected, and then whatever studies need to be done on that cell are then done. So the Buchser lab has a workflow where they combine cell raft with high content imaging. So basically they can introduce mutations, and they can do this by making mutations and then generating what they call isogenic clonal lines, where they're isolating each cell and putting them in dishes and growing them and then performing the analysis. Or they can create a library and a pool of a mixed cell population. So they have ways to introduce different variants. They provide all of these plasmids or other ways to introduce these variants into a cell pool. And the cell pool all gets different variants, or some of them might get the same variant, but at any rate, you get a whole mixed population with different genotypes. These are then put onto the cell raft chips. They're image captured by a high content imaging system. And then he uses a machine learning algorithm to combine a series of phenotypes of the cells in order to try to distinguish them from each other. You get a set of predicted genotypes based on the combination of phenotypes that you find with the imaging. And then you can pick the cells and sequence them within the same workflow called RAFT-seq, so that he can then determine what was in fact the actual genotype that you saw with the specific phenotype. So these cells are all imaged in a single cell individually. So he has done this for a mitochondrial disorder. And this is a preprint that was on bioRxiv that just came out in March. So the table on the left shows the variants that he tested, 5 benign variants and 6 pathogenic variants. He created a mitochondrial, he cultured these cells. On all of these here are the mutant cells, these here, and these are 2 wild type cell lines. And then these are stained with basically what's like the mitochondrial version of lysotracker. It's called mitotracker, and it tracks the number of mitochondria that are present. So as you can see, even in this mitochondrial disorder, you don't see very much difference between the wild type lines and the mutant lines just by looking at them. And that's shown here quantitatively. If you did the mitochondrial count with the mitotracker, you really don't see a difference between the green benign, blue wild type, or red pathogenic lines. There's another thing that feature that he looked for called potential variation, which also didn't really show reliable differences between the pathogenic cohort and the benign cohort of variants. However, using a group of 26 features and something called UMAP, which is a way of reducing the dimensionality of topographically complex structures, his machine learning algorithm was able to generate something that separates pathogenic from benign and wild type variants. And so they ran this through the system, and it was able to show differences between the wild type and the pathogenic lines in both a mixed and a pure population. Here's showing labeled or pure population sorting determination. Here's in a mixed cell population, and then just showing that some of the different alleles performed a little bit differently, but many of them were sortable and were determinable in this workflow. So here you see a randomized assignment where they kind of generated random controls and showed what noise would look like, and then model assignment, and you can see that it performs differently than the controls in the area under the curve and the discrimination of the phenotype. So currently our IDS project, we have moved into a cell line that can be imaged on this cell raft and RAFT-seq platform. The Gurnett Laboratory has generated an IDS knockout cell line in the A549 cells, which is the adenocarcinoma line. And they have generated a GFP LAMP1 reporter construct for transient transfection so that the lysosomes will fluoresce in our system. These have been imaged on the RAFT-seq, and when using the LAMP signal alone, we could confirm that in this cell line too, we weren't able to reliably separate the IDS knockout lines from a wild type control or from a heterozygous IDS line. So down here on the left bottom, you can see the wild type IDS clone and mixed population, and then a full knockout, which is an exon 9 deleted. And then there's a heterozygous cell line with a wild type allele and one knockout allele. The preliminary results that the first run did show that a combination of features was able to discriminate the knockout line from the partial knockout, and which performed very similarly to the wild type clones. Even though when you look at this population, you can see cells are different, but it would be pretty hard to kind of come up with a single feature that would discriminate the knockout from the wild type. So the idea makes a lot of sense. It's kind of like, I can see when you can look at cells and you can see that they might be different, much as you look at people and you can see features in people and patterns that are more than just looking at one thing. It's looking at a combination of things, and sometimes it's hard to describe what those individual things are that you're generating, and how you're figuring out how to tell an affected cell or an affected individual or affected mouse from wild type. But you do it naturally, and the machine learning algorithm can also do this. So our future plans, we've commissioned a set of variant cell lines to serve as our next training set. These lines contain benign, attenuated, and severe variants, and these have been chosen based on published cases where the phenotype is known. We plan to continue to train the RAF-seq imaging model to discriminate based on this training set lines. And then we've have a next set, a test set of variants that we will commission to run that to make sure that the algorithm works. And then we have the ability to validate lines with enzymatic assay and by culturing the isogenic clones by glycosamine and glycan analysis. So that is all. And I'd like to acknowledge the work done by the Christina Gurnett's lab, including Naomi Tayabi, she goes by Majda, and Kevin McCall, and then William Buchser lab, and Jack Bramley in his lab, and in my lab, Soila Sukapolvi and Stephen Lee, and next door to me, Mark Sands. And I'm grateful for the support of the National MPS Society. And we've just learned that our variant test set is going to be funded by the Hope Center for Neurological Disorders and by Washington University's Institute for Clinical and Translational Sciences. And with that, I will end.
Welcome back. What an exciting presentation both of you were able to give. Patty, I was particularly pleased to see that your work has garnered some extra support, additional support to really allow this platform to move forward. So exciting. You gave a wonderful, comprehensive, and it dovetailed really nicely about the need to get these things, these patients phenotyped smoothly and quickly and as comprehensively as possible. And I'd like to open up a question to both of you. So jump in if you think of answers right away. What is the prospect you see for getting this kind of diagnostic up and running in a high throughput way? We're potentially looking at the RUSP getting approved for MPS 2, and of course the window for getting national testing by state, we may be in a 5-year period, but are we potentially looking at a kind of platform that might be available to clinicians within that framework?
So, I mean, our assay is still needs to be refined, and but once it is up and running, there are two potential ways it could be used. I mean, one is if we are able to generate variant libraries at scale, which is the idea, in the A549 cells, like we could in the HAP1 cells, then yes, this could be an easily high scale. In fact, we could potentially generate a lookup table from these experiments, at least by single variants. This doesn't really address the issue of combination of variants or cis variants like Dr. Steet was talking about. Even so, I think the number of individuals who would need this kind of testing for a specific MPS is fairly low, and so This is something where if we aren't able to get a high throughput library generated for a bunch of different variants, one could test this ad hoc on individual variants once the assay is completed.
Yeah.
Excellent.
Yeah, I agree. I mean, ideally, you would love to already have this information in hand, at least some determination of whether these are going to be pathogenic or not as the newborn testing is done. We're making efforts to just make our platform more efficient, but still, you're talking maybe 5 to 6 weeks before we had a good, confident sense of specific activity for a different variant. Whether that's feasible, that certainly gives an answer within a reasonable time frame. But one of the other challenges is, can this be a clinical diagnostic test, and can it be offered as an add-on? And that's, I think, another future challenge for us to consider.
Excellent. But it sounds like the ability to evaluate 2 mutations on the same parental allele, that's something that your system, Rich, really can accommodate more easily, whereas the system you presented, Patty, that's really looking at single changes, polymorphisms. Yeah. Is there anything about the MPS II locus, which has a pseudogene, and there is a potential, I suppose, for strange rearrangements between that and the normal locus? Does this complicate the work that you guys are doing in any specific way?
So I probably lack the full sophistication to answer that. I use the genomic engineering and IPS core at Washington University, and so far they have not encountered an issue making the variants because of the pseudogene, but time will tell.
Yeah, we've had one thing that we've run into is, making the knockout cell line. Using CRISPR, when you get that resulting cell line, there is some adaptation. Patty may be aware of this as well, but I mean, you can get upregulation of other genes that can confound your activity analysis. So that's one challenge with this approach. But once you've got a validated knockout line and you understand it, hopefully addition of the variant forms of these different genes is fairly consistent.
Now, zebrafish are unusual because they often have a duplicated genome. Is the IDS gene present as a single copy in zebrafish, or are there 2 copies?
I think it's just a single copy.
And I'm guessing they don't have the problem of the human pseudogene, so that makes your potential knockouts a little bit more streamlined.
Right, and we are actually using the knockout line of Enrico Moro. And the idea for the going to zebrafish would be if you have a variant that the activity is kind of right at a borderline, or it's not clear from your cell-based testing that it's pathogenic or not, it could be introduced into the zebrafish system and assessed phenotypically. Maybe we're missing something in vivo, in cells that would be apparent in vivo. So it's kind of the last tier. And we would only look at ones that are tricky or, at a borderline or just unclear and need that second level of testing.
Excellent. Patty, your presentation was a great example in my mind, especially for people who may not be steeped in biology and what's happened lately, just where we've gotten to in terms of the complexity of things that we can accomplish. That's a great example of what I call the omics age in science, where we can ask questions that are really 1,000-fold more complicated than we used to. I remember when I started my PhD, we'd spend months just sequencing one gene, and now you can get that kind of comprehensive data in a flash. Do you see any potential for the resources that you are generating, Patty, or yours, Rich, to help drive future diagnostic or drug screen approaches for these disorders?
So I think, yes, the answer is yes. platform that the Buchser lab is using is really very, very powerful because it is an unbiased approach of multiple phenotypes that are combined in various ways by the machine learning algorithms to differentiate groups of cells. So it screams to be used for a therapeutic pipeline. So I think that there is definitely some power in that. I do want to kind of address a little bit the philosophical issue you raised about technology, because the individuals who are deploying these new technologies are not the same individuals who know these diseases, and it is really, really important that medical geneticists and other people who are familiar with these different disorders remain engaged and involved in this revolution that's going on in sequencing, because otherwise, as we've seen, the misdiagnoses of these patients, can have real effects. And, Matthew, you and I and Lauren Clark wrote an editorial for the journal just recently about this, because we have been getting in cases where people are diagnosed and referred for treatment who do not have MPS, because the people at the diagnostic laboratories are deploying the sequencing technologies are, they're very good at that, but they don't understand what they're looking at.
That's a good point. But yes, the tools from both platforms would be cell lines expressing specific variants in these genes, and those would be what you'd want to use to test specific therapies. For instance, if some variants lead to misfolding of an enzyme, would one then be able to use that to test a series of chaperones? So yes, that's one of the benefits longer term, is that you would have these cell-based tools to assess therapy.
I've often been intrigued and enamored of the idea of chaperone in therapy, but there were very few platforms that could address the multiplicity of potential mutations and provide a high throughput approach. I know recently, just this last month, there was a new chaperone developed for MPS 3B that raised background activity 2.5-fold. That was an arduous, designed, empirically structured chaperone. And the sorts of resources you guys are generating could make a platform that would be quite powerful, I think. And Patty, your point about the importance to integrate real clinical knowledge with the diagnostics as they move through newborn screening, et cetera, was so well underscored by the synopsis you gave of that case at the beginning of your talk. This is really critical, and the society works as well as we can with states that are developing these protocols to provide support materials, et cetera, because it's required for each state to set up a pattern of how they're going to test these diseases, their second-tier test, how they're going to develop outreach materials, et cetera. And that's one place the society feels we have a potential impact to reinforce the need for really grounded clinical medical genetic expertise, making sure they're getting GAG testing and not relying simply on genomic predictions. Very good point that you bring up. I also wanted to note that you are both, in addition to receiving MPS funds, you're both members of our scientific advisory board. Patty has been a past chair, and Rich is currently serving on our guidance committee. And I want to take this moment to thank you both for your dedication to the society and to our MPS and ML families.
Thank you.
Yes, thank you.
Any other thoughts that you have? I'm going to check down and see if we have any more questions as we close. And if you don't, I think we'll wind up and get on to the last session.
Great. Thank you so much for having me.
Yes, thank you.
Thank you guys for a wonderful session. It's great to see you, and it's great to hear such good science.
Definitely.
Take care.
Bye-bye.